Assalamualaikum and selamat tengah hari. Okay, so today we're gonna continue our lecture. Okay, so the topic for today is types of bioreactor. Okay, welcome back to your faculty lah. Okay. Okay, actually my aim is actually to have a sharing session with you all because you already go to your field trip last 2 weeks, right? But because of for today it's only a recorded lecture session, so unable to communicate with you all, but never mind, might be we have some time later. Okay, so for today you're gonna learn on the strategies to decide or to choose the bioreactor design, and you're able to know the schematic diagram of each of the bioreactor design, as well as to identify the advantages and disadvantages of each design. Okay, so basically for types of bioreactor for your stage, so I think you are focusing on these 5 different types of bioreactor only, which are stirred tank bioreactor, the pneumatically operated agitated bioreactors, there are 2, airlift and also bubble column bioreactor. Okay, and then immobilized cell bioreactor, membrane bioreactor, as well as the photobioreactor. Okay, so before you go to each of the design, you need to know different design requires different strategies. Okay, so what kind of thing that you need to consider when you are choosing a bioreactor? So this is the thing lah. Okay, so you need to know what kind of microbe that you want to grow, the growth and also the oxygen requirements of each particular microbe, the shear sensitivity or the rheology effects, okay, how to clean or how to sterilize the media, the broth or the bioreactor itself. Does it require light? Is there any foam will be produced? How to prevent the foam from produce? How to heat and how to cool the bioreactor, and also what kind of the material that you are using to fabricate the bioreactor. Okay. Okay, next is actually the step involved in bioreactor. Okay, particularly if you are using bioreactor, you should have the using of shaker flask first to actually cultivate your microbe inside. Okay, once you already cultivate the microbe, you will transfer your microbe into seed tank. Okay, so this seed tank is actually to multiply your microbe. Okay, and then you have the inoculum tank. So in this inoculum tank, you will put your microbe plus with all those broth that contain nutrient and so forth, and then you will further put it inside your large production fermenter or large bioreactor type, so that your product will be formed inside this bioreactor. Okay, so this is the roughly what kind of thing that you need to know, the several steps before you are using the production fermenter or production bioreactor. Okay, so there are 2 classes of bioreactor. We have aerobic and also anaerobic bioreactor. Okay, so this is dependence of the process involved. The aerobic is the one that contain oxygen. Anaerobic is the one that contain without oxygen. Okay, so if you have oxygen in this anaerobic process, it will destroy the microbe. Okay, and then how to classify the bioreactor? You have based on the mode of operation. Okay, the mode of operation of each bioreactor, you have 3. The first one is the batch process, the fed batch process, and also continuous process. Okay, the batch process mean you have one shape flask, you put all your microbe and your nutrient inside, your broth, media, and so forth. You leave it for 1 hour to stir. Okay, and after 1 hour you just take it out, and you take out all the product that you obtain. Okay, but for fed batch, it means that in internal ataupun interval time, you have something that you need to add, but there is no product formation taken out. Okay, for continuous, it means that the inflow and outflow is simultaneous or equal. Means that if you are adding 10 ml of your microbe in 1 hour, so 10 ml of product will be produced in 1 hour as well. Okay, so that is the definition of mode of operation. Okay, so basically the mode of flow of fluids also different. One is a continuous flow or ideally mixed process. Another one using piston flow, which is plug flow bioreactor. Okay, the third one is based on the phases. Okay, you have homogeneous bioreactor that only involve one phases. If you are in liquid form, it only liquid. If you are in solid form, it's only solid process. But for heterogeneous bioreactor, it requires two phases lah, ada solid liquid or liquid gas or gas liquid. Okay, clear? So the classification of bioreactor is based on three: the mode of operation, the mode of the flow of the fluid, and also the phase. And then the classes of bioreactor, you have two: aerobic or anaerobic. Okay. Okay. So without delaying more time, we go to the first type of bioreactor. We have stirred tank bioreactor. Okay. So this is the stirred tank bioreactor that you have in our lab. Okay. So this is the schematic diagram, the basic one. So this is the most important type of bioreactor that I usually use in industry. Why? Because it is low capital and operating cost, and also it is depend largely on the amount of heat to be removed. Okay, you have the stir driven punya agitation method. Okay, you see here, this is the agitator. So you have the motor. Once you on the motor, this agitator will be mixed inside. Dia akan bergerak. Okay, and also you have the baffle. This is the baffle kat tepi ni, the buffer kat tiang tepi ni. So this buffer is actually to prevent the vortex. Okay, vortex is actually the movement of liquid yang sangat laju. Kalau kamu example of if you stir the ice regularly, it will become buffer. Macam ada buffer lah kat tengah tu. Okay, so the buffer is actually not good lah, because it can cause the microbe to be ruptured, because of some microbe is really sensitive to pressure or the shear. Okay, and also it have a high agitation and aeration. Okay, so this agitation and aeration you need to control, because it might cause foaming problem. Okay. Okay, so this is a bioreactor lah. Okay, it might be in cylindrical or curved base, usually using stainless steel. Okay, so it's actually one of the most used bioreactor in worldwide lah, in industry. Okay, so this is the schematic diagram of the stirred tank bioreactor. As I said, it has the motor, it has the sparger. So the gas will be coming from the bottom of your bioreactor. You can see the sparger of the gas at the here. Okay, so this is the baffle to control the mixing agitation process ataupun to prevent the vortex from happen. Okay, this is cooling jacket to control the temperature of your liquid. So the cooling water will be coming from the bottom. It will pass through the jacketed, and also it will go out from the top. Okay, so you have the condenser here, you have the impeller, the blade, and so forth. But I won't go to detail into this blade, because I think you will learn it inside your proses hiliran in your scale up punya lecture. Okay. Okay, the most important thing here is actually the working volume lah. Okay, working volume. Okay, this is a total volume. Here, this is the total volume of your bioreactor. But usually when you want to conduct some fermentation process, the total volume is actually the volume of your bioreactor. But when you want to conduct the fermentation process, make sure you only use 70% of the total volume as your broth. The rest here, you just left it to become head space. Just left it so that any gases that produced will be trapped here. Okay. Okay, so basic feature of bioreactor, you need to make sure that there is agitation system. Agitation for here is impeller lah. Oxygen delivery system here, spudger. Foam control system, you need to have one probe here, add one probe here, so that it can measure the forming of the formation of foam. Okay, temperature control using the cooling jacket. pH control, you need to have the pH control here. Sampling pot, okay, you have to have the sampling pot at the bottom and also at the top. Okay, cleaning and sterilization, how to clean and how to sterilize your bioreactor. And last one is a pot to sum and dump a line, okay, to empty your bioreactor for sterilization process. Okay. Okay, second is pneumatically agitated bioreactor. Okay, for this bioreactor is actually without any mechanical agitation, means that they don't have any impeller. Okay, so the mixing is based on the movement of the air bubble. Okay, if you see here from this schematic diagram, when you purge the oxygen here at the bottom, because of the different pressure, bottom have higher pressure, top have different pressure, so the movement, the air will be moved from the bottom to the top of your column. Once it moves, it will bring together all those microbial cell. So the microbial will go up with this air, so it will move to the top of the column and it will pass through to the left and also here, left and also right of this tank. So the movement will be freely throughout the process. Okay, so during the movement there is mass transfer from your cell from your from your what you call it, from your broth into your cell, as well as from the cell into your broth. Okay, so it have two different three. We have internal loop, external loop, and draft tubes. Okay. Okay, so this is the advantages of an airlift bioreactor. Okay, this is the example of airlift bioreactor lah. Okay, it have low shear because of no mechanical agitation or no impeller. So microbe that sensitive to shear can be used here. Okay, and also it is since there is no agitation, it is easier to sterilize it. Okay, or to cuci guna chemical component lah. Okay, and also the height can be as high as 60 meter. Okay, but once it high, so the pressure at the bottom is higher, so oxygen solubility will be higher at the bottom, so the mass transfer is higher at the bottom lah. Okay, but the disadvantages, the main disadvantages is actually it is difficult to contain consistent kan dia punya carbon source, nutrient source, and also oxygen source throughout the column. Might be the carbon, nitrogen, and oxygen source is higher at the bottom but lower at the top. Okay, it is disadvantages of this airlift lah, and also difficult to separate the gas and liquid. Okay, because if there are any foam is present, if no foam is forming, so it's easier to separate the gas and liquid, because the gas will be at the top here, the liquid will be in this broth. Okay. Okay, bubble column. The differences between bubble column and airlift, actually bubble column they don't have any draft tube. Okay, for airlift they have two. Some other the draft tube will be in the middle of the column, or it will be another external draft tube on the left or on the side of the column. Okay, so that is the differences lah. Means that for bubble column the movement is only based on the anything that you purge from the bottom, but and it will move freely. But for airlift, it will be based on what kind of the draft tube that you are putting in. If the draft tube is on the middle, so the gas will be passed through from the bottom into the top, and it will pass through to the left and the right. But for this one, if the draft tube as external loop, okay, so it will be the gas will be passed through on the top, and it will go to the external loop and coming down, and it will be in the middle again. Okay. So this is example of the airlift bioreactor with external loop, okay, for mammalian cell cultivation. And this is example of the largest industrial fermenter, airlift fermenter, that as high as 200 feet height and 25 feet diameter. This is dia letak kat bawah lah. If they want to use, it will be dia akan tegakkan. Okay, next we have learned the continuous stirred tank and also pneumatically agitated bioreactor. So next is immobilized. Okay, for stirred tank you have the stirrer, agitated or pneumatically agitated bioreactor without any mechanical agitation system, but it have based on the movement of the gas. But for this immobilized cell bioreactor, it means that your cell or your catalyst is being immobilized in support material. Okay, so for this case, okay, you need to consider the mass transfer requirement, the kinetic consideration, as well as the particle characteristic of your bioreactor or your packed bag. Okay, so what is immobilized cell? Immobilized means that the biocatalyst is, or the enzyme is immobilized with insoluble matrix. Okay, so it means that your cell cannot move freely. It will be attached on the surface of your support material. Okay, so this is example. Okay, for immobilized we have lots of things. Okay, one example of bioreactor that using immobilized punya cell is packed bed bioreactor. So this is the image of packed bed bioreactor. You can see the cells are immobilized on large particle, not moved in liquid. Okay, so this is the cell, simple to construct and operate but suffer from blockage. Okay, because it is packed. Pack means dia sangat rapat, dia pack. Okay, so this is the feed coming from the top. This is the effluent stream. Okay, this is the air coming from the bottom. Okay, you can see here air flow in counter current direction to liquid. Liquid coming from the top, air from the bottom. So this is your packing material. The dot one is actually the cell that is already being immobilized ataupun cell that attached to the support material punya surface. Okay, so the formation of product actually occur here lah, kat sini. Okay, product occur here. Okay, so any formation of product or substrate utilization is occur here because cell is here at the top of the porous material. Okay, here trickle back. Okay, for this type of immobilized cell, I show you three example of immobilized bioreactor, which are packed bed bioreactor, which is your immobilized cell is being packed into a bag in this bioreactor. Nampak macam bentuk bag kan? Okay. Second one is trickle back bioreactor, which means your packing materials ni disusun di sini. So your immobilized cell adalah in this bag also, but your feed coming as a trickle. Maksudnya macam direnjis saja on the top layer of your bag, and then it will be trickle down to the flow in the small stream until it pass through all this bag ke bawah. Okay, so this is the simplest reactor type for performing catalytic reaction where gas and liquid are present in the reactor and it is extensively used in processing plant. Maksudnya ada gas kat sini. Ini liquid ke bawah. Okay, gas dia akan ke atas. So that is mass interaction or transfer occur here slowly. Okay. Another one is fluidized bed reactor. Okay, for this fluidized bed reactor, this is the small particle here is actually contain the immobilized cell. So this small particle will be inside your column. Okay, so the feed and also the air will be from the bottom. So it will pass through to the top. So inside here your cell move freely. Because cell move freely, cell to attach to the particle. The particle is move freely. That is it called as fluidized bed bioreactor. Okay, clear? So this method actually maintaining high biomass concentration and also at the same time good mass transfer rate. Good mass transfer tu yang akan memberikan high yield of your product, because the mass transfer, you have the cell tu dia akan react once the nutrient, all those nutrient coming in pass through the membrane. Okay, so that is called as mass transfer rate. Okay, so next is the example of fluidized bioreactor with immobilized cell. So ini example lah. Kamu tengok kat sini ada immobilized cell. So this is usually used in industry. Okay, so this is advantage of immobilized cell. Okay, might be multi-step reaction possible to occur. Okay, but the disadvantages adalah once there are multi-step enzyme reaction, so unwanted side reaction or unwanted product might produce. Yang itu kita tak nak. Okay, and then it is more stable. Why? Because your cell is already encapsulated inside porous material. Okay, high cell density can be employed because you can control the concentration of cell that you akan encapsulated inside immobilized porous material. Okay, and it can be used for a long period of time lah. Okay, and the product can easily remove because your cell is entrapped into something. One is form a product, it will be freely inside your media. So using a suitable downstream processor, you can easily remove the product. Okay, but the disadvantages might be that cell leaking because you encapsulated your cell. So if the porous material pecah, so your cell will be leaked out from your immobilized material. Okay, so this is example of your immobilized cell lah. Okay, so you can see this is the beads. Okay, inside the beads you have the cell, the biocatalyst or the cell that you want to use during your fermentation process. Okay, so this is the methods of immobilization. Okay, you have active immobilization process, passive immobilization process. For active, you have cross-linking, covalent bonding, entrapment in gel. Passive, you have adsorption and colonization. Okay, for active, it means that the what we call it, the microbe, the cell or the immobilization process occur by addition of chemical agent. Okay, but for passive, it will occur naturally. Okay, so this is the process. If you see here, this is the active one, this is the passive one. Okay, so for the active one, you need to, this is the enzyme that you want to add to trap in. So you have to add some a chemical agent, so it will bond each other, so it will form these beads. Okay, but for this one, you have to have you need to add this active support, and you need, this is your cell, so you have to add some chemical agent as well, so it will become attached to each other, so this enzyme won't move freely lah. Okay, and then this one you need to add something, gel or matrix, polymer matrix, that can enough to prevent from this cell to be relieved, but it as well as it allow the penetration of substrate and also the diffusion of product. So the formation of product occur here, but because of this matrix is porous, so your product will be diffused out from this entrapment immobilized cell. Okay, but for encapsulation, as I said, it won't have any addition of active chemical agent, so it just what we call it, the electrostatic interaction between this cell, okay, with the support material will help it to actually attach to each other, to absorb to each other naturally based on the different zeta potential. As well as for this encapsulation, the enzyme will attach to each other and will be encapsulated inside a semi-permeable membrane by its own. Okay, so that is example of how can we do the immobilization process. Okay, Next is membrane bioreactor. So for this membrane bioreactor, it is usually used for treating wastewater. Okay. So how does it perform? Okay, this is your membrane. Okay, membrane bioreactor. So when you add wastewater, so this membrane actually act as a filter to trap all those unwanted dissolved material. So what you get is actually clean water plus with microorganism that you want only based on the sizes. Okay, so usually this one is used um, in bioreactor that are using to, we call it, treat wastewater lah. Okay. So advantages of membrane bioreactor is actually it provide in situ separation process, okay, and also compact to immobilization. For this one, no chemical agent or harsh condition are employed. But the challenge for this membrane bioreactor is actually it is difficult for you to have apa ni, to apa ni, to avoid the cell or membrane become entrapped to the membrane. Kalau dia entrap here, example lah, this bigger size material entrap here, so the flux, the flow distribution is uneven and it become clog here, so dia akan tebal lah kat sini, so might be water pun tak akan pass through. Okay, itu antara challenging lah. Ada banyak lagi, I think you can read again. Okay, and further study lah. Okay, next is photobioreactor. Okay, for photobioreactor is designed for green and blue-green bacteria algae or microalgae, which consider the use of source of light for photoautotroph and also photoheterotroph algae. Okay, so for this type of photobioreactor, you have in tubular punya photobioreactor. Okay, and usually they have two kind of system. They are using a pond ataupun a deep channel culture system with closed circulating loop. Okay, so ini contoh nanti you baca. Saya nak tunjukkan sini. So this is bioreactor, photobioreactor lah. You can see it provide you illumination light. Okay, so this is example of tubular photobioreactor that are using in industry mainly to grow the microalgae. Okay, and this is a flat bioreactor. Nampak flat kan? Yang ni dia bulat-bulat tubular in tube. Ini macam panel solar gitu. Okay, so this is the green fuse closed system algae bioreactor. This is to produce, ini bukan bioreactor, ini contoh untuk production of ethanol. Okay, itu sahaja for today. Okay, a part of this lecture, I will upload also additional note for each different kind of different kinds of bioreactor, as well as some additional video on on different types of bioreactor. So hopefully you will learn more lah. Okay, and also I will attach some quiz, so hopefully you will able to answer the quiz plus with attendance lah, attendance record. Nanti once you dah settle buat quiz tu, you need to scan for your attendance. Okay, sorry guys, because I cannot afford to have face to face punya lecture with you all, and also I cannot afford to have the online in situ punya online lecture with you all. Hopefully this video help you a lot. Okay, see you tomorrow, see you next lecture. Next lecture will be physical lecture lah. Okay, I think that's all from me. Before that, make sure you answer my assignment based on your field trip or ataupun anything, any industry that you are capable of to write up your report. Okay, I think that's all from me today. Thank you everyone. See you later on our next session. Okay, thank you.